Thanks, Dr. Hirschfeld. Thank you so much. It's really a delight to be here and a great honor to be sharing the award this year with my friends and colleagues. I first got interested in bipolar disorder when I was a medical student, and I helped care for a young woman who was admitted to the psychiatric inpatient unit at Johns Hopkins after a suicide attempt. And when she sat down and talked with me, she described how she had suffered from terrible episodes of mania and depression really since her late teens. And that she had been diagnosed with bipolar disorder in her 20s. She said that she would often feel well for months or years at a time when she was taking lithium. But the illness clearly ran in her family, that her father had it, and her grandfather, she thought her brother probably had it as well, and she was worried that her two young children might inherit the illness themselves. And what struck me about her, and what continues to puzzle me about bipolar disorder, I think to this day, is how it could lie dormant for months or years at a time, only to come out in the most disruptive way, taking over someone's thinking, their behavior, and really their entire lives while they're ill. I was also struck by how such a simple thing as lithium, which is really just a salt, could have such a powerful preventive impact on the illness. But most of all, I was puzzled by how it ran in families and why that was. And that's how I really got interested in studying the genetics of the disorder. Now, when I started in the field, which was almost exactly 25 years ago, as a fellow in Dr. Ray DiPaolo's group at Hopkins, we knew very little about why bipolar disorder ran in families. We knew that it certainly could come out in a variety of different ways. This is a classic family study that was done by Elliot Gershon and colleagues, showing that when individuals have the classic form of bipolar disorder, bipolar I disorder, that their first-degree relatives, their children, their siblings, and their parents, have between about a 9-fold increased risk of having bipolar I disorder themselves. But that we also see increased rates of bipolar II disorder, which is a form of the illness with milder elevations of mood, as well as unipolar depression. And we see the same kind of familial clustering if somebody has bipolar disorder, and if they have unipolar depression, we see it, but to much less degree. We also knew back in 1991 that most of the genetic risk for bipolar disorder could be explained in the overall inherited genetics. For example, when we look at twin studies, identical twins who share all of their inherited DNA had about a 70% risk of developing a mood disorder if their co-twin had bipolar disorder, whereas fraternal twins who share only about half of their DNA on average had only about a 30% risk of having a mood disorder if their co-twin had the illness. So this tells us that the heritability of bipolar disorder is something on the order of 90%, and this has been a fairly consistent finding throughout almost a century of twin disorder research. And adoption studies also told us that if we look at adoptees with bipolar disorder, that their risk for the illness tracks through their biological relatives, not their adoptive families. So that bipolar disorder is not an illness that's caused by family upbringing or bad parenting. Let's see, make this go forward. There we go. Now, around that same time, genetic linkage studies started to be successful in identifying genes that cause rare inherited disorders, such as Huntington disease and cystic fibrosis. So a lot of us in the field thought that genetic linkage studies might also be effective for identifying genes involved in common inherited disorders like bipolar disorder. Now, linkage studies work by tracking the inheritance of regions of chromosomes through affected and healthy relatives in a family. And they work very well for single gene disorders. They've probably identified several hundred such genes over the last 25 years. But they don't work so well for psychiatric disorders. And at the time that we began, we didn't know that this was going to be the case, but after about 10 years of work, we found that linkage signals for psychiatric disorders tended to occur in a single family or maybe a couple of families, and that other families seemed to have rather different linkage signals. And even when we could find more consistent signals, as we did in chromosomes 18, chromosome 6, some other regions, in the end we could not find any genes that explained the linkage signals in those families. So after about 10 years of work on linkage studies, we really were left with the situation where we were searching under the lamppost. Think of this as a linkage finding, but the keys were still out of reach. And the breakthrough that really opened things up for this field were genome-wide association studies. So for the first time with genome-wide association technology, we were able to illuminate the entire genome space through the use of thousands and thousands of markers that could be genotyped in an automated way in a single experiment, making it much more likely that we could find the keys involved. Now, when we published the first genome-wide association study of bipolar disorder back in 2007, we were amazed to find that there were actually dozens of markers that were associated with the illness in two completely independent samples. We expected we might find one or two. We were also surprised to find that each marker was very common, actually more common than bipolar disorder itself in the population, which is about 1 or 2%. Many of the markers mapped near identified genes, but the genes were unfamiliar, and they weren't the kinds of genes that we had expected to find based on what we thought we knew about the biology of bipolar disorder and its treatment. We could also see that each individual marker conferred only a very small risk, and it seemed that many markers came together in an individual to confer their risk. And we recognize this as an example of what's called the polygenic threshold model that had been described actually by theoretical geneticists back in the 1960s. Now, under the polygenic threshold model, it's assumed that everyone carries some genetic risk factors, and that these are distributed normally in the population. So some people have a very small dose, some people have a larger dose, and that, on average, people are not affected. However, there's a portion of the population, shown here in blue, that actually inherits a larger dose of genetic risk factors. And those individuals might develop symptoms and end up being considered affected in a genetic study. So you can think of, and this shows on the line, how the dose of the risk alleles increases as you move through this distribution in the population. Now, you can think of genes in this polygenic model almost as if they're grains of sand piling up in a sand dune. When the dune gets high enough, through gravity alone, sand topples over the top and creates those sharp ridges that we see when we go to the beach. This sort of ridge really corresponds to the threshold in the polygenic threshold model. So at this point, we thought that perhaps we had made a start, but we were puzzled by the results that we found. And we were concerned that perhaps we had gotten it completely wrong. We went from having no genes involved in bipolar disorder to what seemed like having too many. The genes were pointing to genes, the markers were pointing to genes that we really didn't expect to find. The methods that we were using were still pretty new and unproven, and our initial results really didn't live up to the kinds of stringent statistical significance that we would later learn was very important in genome-wide association studies. So our worry was this just another false start? We were so delighted when, about a year later, a much larger genome-wide association study was published in a much larger sample. And this was co-authored by Dr. Pamela Sklar, who very appropriately is another Colvin Prize winner today, and you'll be hearing from her later on. What was so amazing to us about this study was that some of the same genes that we had identified in our study were showing up in this study, in particular this gene ANC3. They were not the same markers. They were not exactly the same risk alleles. But the implication was that perhaps for the first time we were on the right track in being able to find reproducible genetic markers for bipolar disorder. And I can't emphasize enough how important it was for the field at that time for us to finally have the sense that after years of what seemed like stumbling in the dark, we finally had an approach, a scientific method that could begin to unravel the genetics of this disorder and other psychiatric disorders as well, and that we finally had a solid starting point from which to go forward. So where do we stand now? Well, as of last month, there are now at least 25 genetic markers for bipolar disorder that have been identified in genome-wide studies, and these all reach the highest levels of statistical significance. They point to multiple different genes, and as we saw in the initial studies, each marker confers only a very small risk, about 1 or 2% increased risk, and so multiple markers have to come together in each individual to really explain risk. We think probably that this is only a portion of all the markers that will be found as sample sizes grow. And what we're seeing is that the story of the genetics of bipolar disorder is not a story of one gene, not a story of a few genes, but actually a story of many genes coming together. We can even begin to estimate how genes come together to influence risk in individual cases. And we do that by tallying up the risk variance that a person may carry. And when we do that, comparing people with low allele burden to those with high allele burden, we find that the risk rises pretty steeply with the burden of risk variance. And that if we compare individuals at the highest end to those at the lowest end of that risk allele spectrum, they have about a 4-fold increased risk of developing bipolar disorder themselves. Now remember that just having a first-degree relative, a parent or a sibling with bipolar disorder, increases your risk about 10 or 15-fold. So we're still not able to fully account for why bipolar disorder runs in families, but we're starting to make significant inroads in our understanding. Now, we've also used genome-wide association methods to ask the question of whether genetic markers actually can influence response to treatment in bipolar disorder. Now, the first thing we've looked at is lithium. Lithium is still the most effective drug for preventing episodes of mania and depression in bipolar disorder, but it doesn't work for everyone. And we reasoned that if we had a way to use genetic markers to identify people who were unlikely to respond to lithium, we might save them the time and trouble of trying a drug that ultimately wouldn't work for them and be able to move on more quickly to some of the alternative treatments that are available. Now, in order to do this, we realized we needed to really have a very large sample, so we put out a call around the world to centers who were willing to donate DNA samples and had information on lithium response in people with bipolar disorder. We were able to put together about 3,000 samples for the initial study, and we're hoping to actually have another larger study down the road. In this initial analysis, we found that there were a group of markers on chromosome 21 that were associated with lithium response in this sample. Now, this region doesn't actually contain any genes that encode proteins, but it has what are called long non-coding RNA genes. We don't really understand very much what they do, but there's initial evidence suggests that long non-coding RNAs play a role in regulating other genes and coordinating the responses of those genes, particularly in the brain during development. Now, when we looked at these same markers in an independent sample that was followed over time by our collaborator, John Kelso, what we found is that individuals who had the good response allele, who were followed for up to 120 weeks on lithium, had a slower rate of relapse than individuals who carried the bad response allele. Now, it turns out that individuals in this group with the bad response allele, that's only about 5% of the population. So we don't think that these markers are likely to be very useful for clinical decision making yet. But they do begin to give us some hints on the ways in which lithium influences gene expression and give us grounds to hope that as the sample size grows, we might actually be able to find additional markers that could, taken together, find a role in clinical decision making. Now, I want to point out that this analysis was done by Liping Hu, who's a fellow in my group who won a NARSAD Young Investigator Award in 2013 for Genomewide Association Studies. And it was led by Dr. Thomas Schulze, who also is receiving a Colvin Prize today, and who I'm sure will tell you more about this study when he speaks in just a few minutes. So where do we stand now? Well, I've told you how genome-wide association studies have led us to many common genetic markers for bipolar disorder, more than we ever expected, and actually that we expect more will come down the road. That these findings are robust and reproducible from sample to sample, unlike what we had in the era of linkage studies. That added together, these markers can confer a substantial risk for illness in people who have the highest dose of risk alleles, but still not as much as having a first-degree relative with the illness. We think there are many more markers yet to be found, so the genetic story of bipolar disorder, which is already complex, will probably get more complex as we learn more about the genetic markers involved. What I haven't told you, though, is how we can begin to move from these genetic markers to some biological understanding of bipolar disorder. Because the real point of genetic studies, really, is to understand what causes uninherited illness, and through insight into that causation, to come up with better ways to diagnose, treat, and maybe ultimately prevent the illness. Now, it turns out that genetic markers are not the same thing as genes. So one of the big challenges we face now in the field is to learn how to link these markers to individual genes, and how to build the genes together into coherent biological stories. We are only beginning on this journey now, but I'll give you some clues as to what we're doing in our lab, and you'll probably hear more about other related work later today. So one approach to this problem is actually to look at gene expression in the brain. Remember, genetic markers only tell us about genetic variation. They don't tell us about the genes that are changed. It's possible that disorders like bipolar illness actually result from dysregulation of the turning on and off of genes in the brain during development and in response to life experiences. So if we can begin to look at which genes have differences in their expression in the brain, that may give us some clues as to some of the underlying biology. We did this in a pilot study in a sample of individuals who had donated their brains to research before death, who had been diagnosed with bipolar disorder in life. And we had a set of comparison individuals that we were able to match on gender and age who had died from other illnesses not related to bipolar disorder. And when we looked at expression in this part of the brain, the dorsolateral prefrontal cortex, which has been heavily studied across a range of psychiatric disorders, we found that there were actually many differentially expressed genes, more than 3 dozen, in fact. and that when we put those genes together with information that we had from genome-wide association studies, we found that some of the genes that were downregulated were also enriched for association signals, and that was also true of some of the genes that were upregulated in the brains of people with bipolar disorder. And now these genes were not a random subset, but actually if we assigned the genes to their known function and began to look for themes, what we found is that among the genes that were expressed differently in the brains of people with bipolar disorder, and which had signals that had also come out in genome-wide association studies, that there were four big themes biologically that those genes shared: ion bonding, transmembrane transport, nucleoside triphosphatase activity, and GTPase binding. Now, most of these terms are not going to be very familiar to you, but one thing that we recognized when we saw these is that these are all essential functions in the ways that neurons talk to one another, signal to one another in the brain. And then therefore, it supports the idea, which much other research has also pointed toward, that bipolar disorder comes from some kind of a failure or malfunction in the way that neurons communicate with each other in the brain. Now, it turns out these genes also don't act in isolation, but are actually correlated with other genes in what are called CoExpression modules. These are the modules that we were able to identify in the study I just described to you. Each one of these circles represents a different gene, and they're colored in the ways in which they correlate with other genes in their module. When we looked at this in this way, one module in particular, shown here in green, stuck out both for how many differentially expressed genes it contained and how strong of an association signal those genes had in earlier genome-wide association studies. And we saw some of the same kinds of signals that we had seen in the individual gene analyses, but now we were able to actually put it together into a bigger picture and point to something called the postsynaptic density. And this was very interesting because it had already been known that the postsynaptic density is actually a very vital structure in the brain. It's a collection of proteins that exist at the receiving end of the synapse, that space between neurons where they communicate, and which is critically important in modulating the crosstalk between neurons and other brain cells. And you can see in this figure, you can get it to appear, there we go, that there were actually a number of proteins from the postsynaptic density whose controlling genes were implicated by our study. So this suggests that perhaps the postsynaptic density is part of the complex causal story of bipolar disorder, and that drugs that target proteins within the postsynaptic density could be promising new treatments. But obviously we have a long way to go before we can say that with any certainty. Now, I talked about the importance of cell-cell signaling and neuronal communication in the brain. And one of the big challenges we faced in the field of neurobiology is how difficult it is to study the brain in action in living human beings. Imaging studies give us some clue of that, and animal model studies give us another, but obviously animals don't suffer from bipolar disorder, don't suffer from schizophrenia, and therefore don't serve as great models for the kinds of illnesses we're trying to understand. Also, I've told you how many genes contribute to diseases like bipolar disorder, so simply knocking out one gene in a mouse or a rat may not give us much insight into how these genes act together to determine risk. Thanks to a new technology, though, known as induced pluripotent stem cell reprogramming, it's now possible for us to collect a tissue sample from a donor, either a skin biopsy or a blood sample, grow these in the laboratory, and then use a methodology that actually allows us to turn them back into, essentially, stem cells that can differentiate into any body tissue. We can then train those cells to go down a neural lineage and eventually be differentiated into neurons or other brain cells. And this is such an important technology because these cells actually capture the full genetic risk allele spectrum in individuals who donated the tissue. So it allows us to study the impact of many hundreds of genes acting together in a particular cell. Get this done. There we go. And some of the kinds of things we can do with induced pluripotent stem cell technology is we can differentiate the cells into neurons in the laboratory and use a technique known as immunofluorescence to actually visualize important proteins and structures. And this is an axon, and you can see how it is expressing in green these postsynaptic density proteins that I talked about in our earlier brain expression study. We can also use a method known as calcium imaging to actually look at the way in which ions, calcium ions, flow in and out of neurons while they fire and signal to one another. Let's see if we can get that to come on here. So we're going to go with this calcium imaging movie. And you can see as the neurons light up in that bright green color, that's actually a sign of calcium moving in and out of the cells as they fire and signal to one another. Now this is great, but actually it's now possible even to go a bit further and grow groups of neurons within individual wells in a culture plate and then monitor their crosstalk directly on what's called a microelectrode array. And as this movie runs, each one of these is a cell in the culture dish, and there are groups of neurons in there that we've grown from people with bipolar disorder and without. And each time you see a blue or a red flash, that means the neurons are firing. And you can see that they don't fire randomly. They fire in groups. And the time it takes for the signal to spread throughout each well, maybe I'll ask you to run that one more time. The time it takes for each signal to spread throughout the well is actually a measure of how well connected these neurons are at the postsynaptic density and other structures. We can use this system actually to screen large sets of molecules as potential new therapies, and then try them out in animals and other systems before we move back into the clinic. Okay, so I hope I've given you a demonstration of the enormous progress that's been made in the genetics of bipolar disorder over the last 10 years, after what was initially a very long and difficult process to find the right methods for understanding this illness. We see that bipolar disorder genetics are certainly complex, but they're not intractable. Our next challenge is to translate genes into biology, and I've shown you how we can integrate GWAS with gene expression studies, how we can begin to model cellular pathology using systems based on iPSC. And I haven't talked about the importance of rare alleles, but that might be another way that we can tag genes of larger effect in bipolar disorder. The goal of all these studies is to reduce the suffering of people with bipolar disorder and to develop new ways to treat the illness and maybe ultimately down the road to prevent it. So I want to thank the BBRF for supporting my work and the work of my fellows over many years. It really has been an enormous support for what we do. And I also want to acknowledge our other funding sources, but folks in my lab and our many collaborators. And with special thanks to all of our research study participants who participate in this research without any immediate personal benefit, but because they're hoping to contribute to furthering the field. Thanks very much. We have time for a question or two?
Yeah, we'll get maybe one question. We're running a little bit behind if you got. Yeah, here's just the one. Yes, ma'am? Oh, I thought we had, isn't it? Does somebody have a question here? Well, yeah.
Is there any correlation between bipolar, Parkinson, and drug addiction?
Can you say it one more time? I didn't quite get the last half of that.
Drug addiction.
Oh, and drug addiction. Okay. The question is there a correlation between bipolar disorder and drug addiction? It's certainly the two conditions occur together in the same individuals more often than we'd expect by chance. What we don't know is whether that reflects a shared genetic causation or whether one occurs as a complication of the other. There are actually some studies underway now that are looking explicitly at that question and might give us a better handle on that chicken and egg problem, which I think comes up not only for bipolar disorder, but for a lot of the major mental illnesses.
Okay. I think we need to.
Okay.
Thank you very much, Russ.